Ribonucleic acid is a family of large biological molecules that perform multiple vital roles in the coding, decoding, regulation, and expression of genes. Together with DNA, RNA comprises the nucleic acids, which, along with proteins, constitute the three major macromolecules essential for all known forms of life. Like DNA, RNA is assembled as a chain of nucleotides, but is usually single-stranded. Cellular organisms use messenger RNA to convey genetic information that directs synthesis of specific proteins, while many viruses encode their genetic information using an RNA genome. Some RNA molecules play an active role within cells by catalyzing biological reactions, controlling gene expression, or sensing and communicating responses to cellular signals. One of these active processes is protein synthesis, a universal function whereby mRNA molecules direct the assembly of proteins on ribosomes. This process uses transfer RNA molecules to deliver amino acids to the ribosome, where ribosomal RNA links amino acids together to form proteins. Comparison with DNA. The chemical structure of RNA is very similar to that of DNA, but differs in three main ways. Unlike double-stranded DNA, RNA is a single-stranded molecule in many of its biological roles and has a much shorter chain of nucleotides. However, RNA can, by complementary base pairing, form intra-strand double helixes, as in tRNA. While DNA contains deoxyribose, RNA contains ribose. These hydroxyl groups make RNA less stable than DNA because it is more prone to hydrolyses. The complementary base to adenine is not thymine, as it is in DNA, but rather uracil, which is an unmethylated form of thymine. Like DNA, most biologically active RNAs, including mRNA, tRNA, rRNA, snRNAs, and other non-coding RNAs, contain self-complementary sequences that allow parts of the RNA to fold and pair with itself to form double helices. Analysis of these RNAs has revealed that they are highly structured. Unlike DNA, their structures do not consist of long double helices but rather collections of short helices packed together into structures akin to proteins. In this fashion, RNAs can achieve chemical catalysis, like enzymes. For instance, determination of the structure of the ribosome an enzyme that catalyzes peptide bond formation revealed that its active site is composed entirely of RNA. Structure. Each nucleotide in RNA contains a ribose sugar, with carbons numbered 1 foot through 5 feet. A base is attached to the 1 foot position, in general, adenine, cytosine, guanine, or uracil. Adenine and guanine are purines, cytosine and uracil are pyrimidines. A phosphate group is attached to the 3 feet position of one ribose and the 5 feet position of the next. The phosphate groups have a negative charge each at physiological pH, making RNA a charged molecule. The bases form hydrogen bonds between cytosine and guanine, between adenine and uracil and between guanine and uracil. However, other interactions are possible, such as a group of adenine bases binding to each other in a bulge, or the GnRA tetraloop that has a guanine-uroadenine base pair. An important structural feature of RNA that distinguishes it from DNA is the presence of a hydroxyl group at the 2 feet position of the ribose sugar. The presence of this functional group causes the helix to adopt the A-form geometry rather than the B-form most commonly observed in DNA. This results in a very deep and narrow major groove and a shallow and wide minor groove. A second consequence of the presence of the 2-hydroxyl group is that in conformationally flexible regions of an RNA molecule, it can chemically attack the adjacent phosphodiester bond to cleave the backbone. RNA is transcribed with only 4 bases, but these bases and attached sugars can be modified in numerous ways as the RNAs mature. Pseudoadenine, in which the linkage between uracil and ribose is changed from a Ca-Euro-N bond to a Ca-Euro-C bond, and ribotimidine are found in various places. Another notable modified base is hypoxanthine, a deaminated adenine base whose nucleoside is called inosine. Inosine plays a key role in the wobble hypothesis of the genetic code. There are more than 100 other naturally occurring modified nucleosides, The greatest structural diversity of modifications can be found in tRNA, while pseudoadenine and nucleosides with 2-O-methyl ribose often present in rRNA are the most common. The specific roles of many of these modifications in RNA are not fully understood. However, it is notable that, in ribosomal RNA, many of the post-transcriptional modifications occur in highly functional regions, such as the peptidyl transferase center and the subunit interface, implying that they are important for normal function. The functional form of single-stranded RNA molecules, just like proteins, frequently requires a specific tertiary structure. The scaffold for this structure is provided by secondary structural elements that are hydrogen bonds within the molecule. This leads to several recognizable domains of secondary structure like hairpin loops, bulges, and internal loops. Since RNA is charged, metal ions such as Mg2+ are needed to stabilize many secondary and tertiary structures. Synthesis of RNA is usually catalyzed by an enzyme RNA polymerase using DNA as a template, a process known as transcription. Initiation of transcription begins with the binding of the enzyme to a promoter sequence in the DNA. The DNA double helix is unwound by the helicase activity of the enzyme. The enzyme then progresses along the template strand in the 3A1 registered trademark to 5A1 registered trademark direction, synthesizing a complementary RNA molecule with elongation occurring in the 5A1 registered trademark to 3A1 registered trademark direction. The DNA sequence also dictates where termination of RNA synthesis will occur. Primary transcript RNAs are often modified by enzymes after transcription. For example, a poly, A, tail and a 5' cap are added to eukaryotic pre-mRNA and introns are removed by the spliciosome. There are also a number of RNA-dependent RNA polymerases that use RNA as their template for synthesis of a new strand of RNA. For instance, a number of RNA viruses use this type of enzyme to replicate their genetic material. Also, RNA-dependent RNA polymerase is part of the RNA interference pathway in many organisms. Types of RNA. Overview. Messenger RNA is the RNA that carries information from DNA to the ribosome, the sites of protein synthesis in the cell. The coding sequence of the mRNA determines the amino acid sequence in the protein that is produced. Many RNAs do not code for protein, however. These so-called non-coding RNAs can be encoded by their own genes, but can also derive from mRNA introns. The most prominent examples of non-coding RNAs are transfer RNA and ribosomal RNA, both of which are involved in the process of translation. There are also non-coding RNAs involved in gene regulation, RNA processing and other roles. Certain RNAs are able to catalyze chemical reactions such as cutting and ligating other RNA molecules, and the catalysis of peptide bond formation in the ribosome. These are known as ribosomes. In translation, messenger RNA carries information about a protein sequence to the ribosomes, the protein synthesis factories in the cell. It is coded so that every 3 nucleotides correspond to 1 amino acid. In eukaryotic cells, once precursor mRNA has been transcribed from DNA, it is processed to mature mRNA. This removes its intrans- non-coding sections of the pre-mRNA. The mRNA is then exported from the nucleus to the cytoplasm, where it is bound to ribosomes and translated into its corresponding protein form with the help of tRNA. In prokaryotic cells, which do not have nucleus and cytoplasm compartments, mRNA can bind to ribosomes while it is being transcribed from DNA. After a certain amount of time, the message degrades into its component nucleotides with the assistance of ribonucleases. Transfer RNA is a small RNA chain of about 80 nucleotides that transfers a specific amino acid to a growing polypeptide chain at the ribosomal site of protein synthesis during translation. It has sites for amino acid attachment and an anticodon region for codon recognition that binds to a specific sequence on the messenger RNA chain through hydrogen bonding. Ribosomal RNA is the catalytic component of the ribosomes. Eukaryotic ribosomes contain 4 different rRNA molecules, 18S, 5.8s, 28s and 5s are RNA. 3 of the rRNA molecules are synthesized in the nucleolus, and 1 is synthesized elsewhere. In the cytoplasm, ribosomal RNA and protein combine to form a nucleoprotein called a ribosome. The ribosome binds mRNA and carries out protein synthesis. Several ribosomes may be attached to a single mRNA at any time. Nearly all the RNA found in a typical eukaryotic cell is rRNA. Transfer messenger RNA is found in many bacteria and plastids. It tags proteins encoded by mRNAs that lack stop codons for degradation and prevents the ribosome from stalling. Regulatory RNAs. Several types of RNA can downregulate gene expression by being complementary to a part of an mRNA or a gene's DNA. MicroRNAs are found in eukaryotes and act through RNA interference, where an effector complex of mRNA and enzymes can cleave complementary mRNA, block the mRNA from being translated, or accelerate its degradation. While small interfering RNAs are often produced by breakdown of viral RNA, there are also endogenous sources of cRNAs. cRNAs act through RNA interference in a fashion similar to mRNAs. Some mRNAs and cRNAs can cause genes they target to be methylated, thereby decreasing or increasing transcription of those genes. Animals have pi-interacting RNAs that are active in germline cells and are thought to be a defense against transposons and play a role in gametogenesis. Many prokaryotes have CRISPR-RNAs, a regulatory system similar to RNA interference. Antisense RNAs are widespread. Most downregulate a gene, but a few are activators of transcription. One way antisense RNA can act is by binding to an mRNA, forming double-stranded RNA that is enzymatically degraded. There are many long non-coding RNAs that regulate genes in eukaryotes. One such RNA is XIST, which codes one X chromosome in female mammals and inactivates it. An mRNA may contain regulatory elements itself, such as riboswitches, in the 5' untranslated region or 3' untranslated region. These regulatory elements regulate the activity of that mRNA. The untranslated regions can also contain elements that regulate other genes. In RNA processing, many RNAs are involved in modifying other RNAs. Introns are spliced out of pre-mRNA by spliceosomes, which contain several small nuclear RNAs, or the introns can be ribosomes that are spliced by themselves. RNA can also be altered by having its nucleotides modified to other nucleotides than A, C, G and U. In eukaryotes, modifications of RNA nucleotides are in general directed by small nucleolar RNAs, found in the nucleolus and cagal bodies. SNRNAs associate with enzymes and guide them to a spot on an RNA by base pairing to that RNA. These enzymes then perform the nucleotide modification. rRNAs and tRNAs are extensively modified, but snRNAs and mRNAs can also be the target of base modification. RNA can also be methylated. RNA genomes, like DNA, RNA can carry genetic information. RNA viruses have genomes composed of RNA that encodes a number of proteins. The viral genome is replicated by some of those proteins, while other proteins protect the genome as the virus particle moves to a new host cell. Viroids are another group of pathogens, but they consist only of RNA, do not encode any protein and are replicated by a host plant cell's polymerase. In reverse transcription, reverse transcribing viruses replicate their genomes by reverse transcribing DNA copies from their RNA. These DNA copies are then transcribed to new RNA. Retrotransposons also spread by copying DNA and RNA from one another, and telomerase contains an RNA that is used as template for building the ends of eukaryotic chromosomes. Double-stranded RNA. Double-stranded RNA is RNA with two complementary strands, similar to the DNA found in all cells. dsRNA forms the genetic material of some viruses. Double-stranded RNA such as viral RNA or cRNA can trigger RNA interference in eukaryotes, as well as interferon response in vertebrates. Key discoveries in RNA biology. Research on RNA has led to many important biological discoveries and numerous Nobel Prizes. Nucleic acids were discovered in 1868 by Friedrich Miescher, who called the material nuclein since it was found in the nucleus. It was later discovered that prokaryotic cells, which do not have a nucleus, also contain nucleic acids. The role of RNA in protein synthesis was suspected already in 1939. Severo Ochoa won the 1959 Nobel Prize in Medicine after he discovered an enzyme that can synthesize RNA in the laboratory. However, the enzyme discovered by Ochoa was later shown to be responsible for RNA degradation, not RNA synthesis. In 1956, Alex Rich and David Davis hybridized two separate strands of RNA to form the first crystal of RNA whose structure could be determined by X-ray crystallography. The sequence of the 77 nucleotides of a yeast tRNA was found by Robert W. Holley in 1965, winning Holley the 1968 Nobel Prize in Medicine. In 1967, Carl Oes hypothesized that RNA might be catalytic and suggested that the earliest forms of life could have relied on RNA both to carry genetic information and to catalyze biochemical reactions an RNA world. During the early 1970s, retroviruses and reverse transcriptase were discovered, showing for the first time that enzymes could copy RNA into DNA. For this work, David Baltimore, Renato Del Becco and Howard Temin were awarded a Nobel Prize in 1975. In 1976, Walter Fiers and his team determined the first complete nucleotide sequence of an RNA virus genome, that of bacteriophage MS2. In 1977, introns and RNA splicing were discovered in both mammalian viruses and in cellular genes, resulting in a 1993 Nobel to Philip Sharp and Richard Roberts. Catalytic RNA molecules were discovered in the early 1980s, leading to a 1989 Nobel Award to Thomas H. and Sidney Altman. In 1990 it was found in petunia that introduced genes can silence similar genes of the plant's own, now known to be a result of RNA interference. At about the same time, 22 NT-long RNAs, now called microRNAs, were found to have a role in the development of C. elegans. Studies on RNA interference gleaned a Nobel Prize for Andrew Fire and Craig Mello in 2006, and another Nobel was awarded for studies on transcription of RNA to Roger Kornberg in the same year. The discovery of gene-regulatory RNAs has led to attempts to develop drugs made of RNA, such as cRNA, to silence genes. See also, DNA, RNA and proteins, the three essential macromolecules of life, references. External links, RNA World Website Link Collection, Nucleic Acid Database Images of DNA, RNA and complexes. tRNA, a game forming RNA by pairing bases. RNA calculators.